Hello everyone, welcome to today's session. We are starting our journey into microbiology. Don't worry, I'm not going to make it boring with heavy definitions and endless theory. Instead, we'll learn it in a simple way. So, let's start with bacteriology first. Okay, growth of bacteria. Bacteria is replicated by binary fission. This is important one. That means it splits itself into half. So, that is binary fission. Reproduce rapidly at an exponential rate under favorable condition. Favorable conditions means nutrients, enzymes, and temperature, etc., required for the growth of the bacteria. When grow in culture media, a predictable pattern of growth is seen. So let's learn bacterial growth curve. So there are four phases in our bacterial growth curve. So the first one is lag phase, then log phase or exponential phase. The third one is stationary phase, and the fourth one is death phase. So the first one is lag phase, and the second one is the log phase. You don't have to confuse between these. So always remember lag is the first one and log is the second one. Okay. So lag phase means adjustment to nutrients and enzyme synthesis. The log phase or exponential phase is the area where exponential growth is seen. So this area is used for biochemical and susceptibility tests. The third one is stationary phase. Stationary phase where nutrients get depleted and waste products are increased. So the bacteria start to die. So it is very important that during stationary phase, we have to shift them into another culture media so that the bacteria don't die. Okay. And the death phase is the phase where all the bacteria die. And the next one is factors affecting the bacterial growth curve. First one is psychrophiles. Psychrophiles means they are usually chill lovers and they grow at the temperature of 15 to 20 degrees Celsius. And the example is Achromobacter, Listeria, and Flavobacterium. Psychrotrophs or you have to remember this. Psychrophiles mean they are usually chill lovers. Okay. And their temperature is 15 to 20 degrees Celsius. Next is psychrotrophs or psychrotolerance. Their temperature range is 20 to 30 degrees Celsius. 0 to 7 degrees Celsius is rare, so you don't have to remember it. Usually it is 20 to 30 degrees Celsius. And the example is Moraxella, Vibrio, and Pseudomonas. Mesophiles, this one is the important class because the temperature range is 25 to 40 degrees Celsius. You know that our body temperature is 37 degrees Celsius. So that comes under this category. So majority of our pathogenic bacteria comes under this. Next is thermophiles. Thermophile is 40 to 80 degrees Celsius. The example is Bacillus stereothermophilus. This is usually used in autoclaving to check whether the sterility has been achieved or not. So the questions usually come, which bacteria is used in autoclaving? So the answer is Bacillus stereothermophilus. The other class is hyperthermophiles. They can survive but cannot reproduce. Example is Pyrococcus furiosus. The next one is gaseous requirement. There are aerobes, capnophilic, and microaerophilic, facultative anaerobes, obligate anaerobes, and aerotolerant anaerobes. So I have prepared a mnemonics for all this and which will make your study easy. So I will explain that mnemonics and I'll teach you that in the later classes or next classes. So first, let's go through this. Obligate aerobes means requires oxygen, and the example is Mycobacterium, Pseudomonas, and Bacillus. Capnophilic means they require carbon dioxide. You can see that in capnophilic there is a letter C, and in carbon dioxide also there is a letter C. So you can remember it this way. Capnophilic requires carbon dioxide for the growth. An example is Neisseria gonorrhoeae and Campylobacter. The next is microaerophilic, which requires very low oxygen. And the example is Campylobacter and Helicobacter. Campylobacter and Helicobacter is both capnophilic and microaerophilic. So that is kind of important. And next is facultative anaerobes. They can survive with or without oxygen. And the example is most bacteria, that is enterobacteriae. Next is obligate anaerobes. They can't survive in oxygen. They only survive without oxygen. And the example is actinomycetes, bacteroids, and Clostridium. And other is aerotolerant anaerobes. They can survive in oxygen, but only for some time. The example is Clostridium tertium. The next one is nutrition and pressure. That is not much important. The salt concentration is also not much important. You have to remember these halophiles and non-halophiles. So halophiles means 5 to 20% NaCl. They're usually Vibrio cholerae, and non-halophiles means no salt, that is E. coli. Another one is radiation, and the next is pH. pH means neutrophils. Neutrophils means the neutral pH. They are the ones that cause mostly all diseases. Mm, that is E. coli, Salmonella, Staphylococcus. Sorry, not mostly, all diseases. Most diseases are caused by neutrophils. So the example is E. coli, Salmonella, and Staphylococci. So next is gram staining. This one is important. The difference between gram positive and gram negative. How can we identify whether they are gram positive or gram negative? First of all, there is a staining method is the best way to identify it. That is, if it's gram positive, then it is purple or violet color. And if it's gram negative, then it's red or pink color. But apart from that, we can make out by the peptidoglycan layer. In gram positive, the peptidoglycan layer is very thick. You can see that in gram positive, the peptidoglycan layer is very thick. But in gram negative, you can see this is the peptidoglycan layer in gram negative. So it is very thin, right? So this is how we differentiate gram positive and gram negative. So you always remember that in gram positive, the peptidoglycan layer is thick, but in gram negative, it is thin. So the questions may come this way. How can we identify the gram positive bacteria? And if there is peptidoglycan in the options, you have to click that option. Okay. The another one is teichoic acid. Teichoic acid is an important one in gram positive. You can remember it this way. Positive people always drink tea. Tea for teichoic acid. So gram positive. Gram positive. Positive people always drink tea. Teichoic acid, tea. Okay. The other one is in gram negative. There is a lipopolysaccharide. There is a lipopolysaccharide. That is also a differentiating factor. So the two things that differentiate gram positive and gram negative is the first one, the important one is peptidoglycan layer. In gram positive, the peptidoglycan layer is thick, and in gram negative, The bacterioglycan layer is thin. Okay. Okay. So now let's go through gram staining. Only these steps are important. The primary stain is crystal violet. The modern is grams iodine. Decolorizer is alcohol or acetone. And the counter stain is carbon fusion. The carbon fusion is the counter stain, or saffronin also is the counter stain. Okay. The primary stain is crystal violet and the counter stain is carbofusion or saffronin. I have not written it, but saffronin is also the counter stain. The modern is grams iodine and the decolorizer is alcohol or acetone. Next, this is gram positive bacteria and gram negative. I have prepared another flow chart to make everything easier. So let's skip it for now. Uh, let's go through special stains. The first one is Wright's Giemsa stain. The rapid method is for malarial parasite and the slow method is for spirochetes. Albert stain. Albert stain is to identify the metachromatic granules. Metachromatic granules means colorful granules that are seen mainly in Corynebacterium diphtheriae. You can remember this way: Albert wearing colorful t-shirt. Sorry, Albert wearing colorful t-shirt. So Albert stain is used to identify metachromatic granules. That is colorful granules. Albert's wearing colorful shirt. So sorry. Next is Loeffler's methylene blue, that is LMB stain, that is usually used for Hemophilus, influenza, Neisseria, meningitis, PMN cells in diarrheal disease, and stool demonstration. Next is acidic orange stain. Acidic orange stain is usually used for fluid exudates, that is T. vaginalis, yeast cells, clue cells in vagina, blood culture. Okay. Auramine phenol stain is usually a fluorescent stain. Okay, there is two types, one and two. The one is used for Mycobacterium and the two is used for Cryptosporidium. Both are fluorescent stains. Okay, next is NMB stain, that is new methylene blue stain. It is used to know whether bacteria is present or not. It is used to know, just to know whether the bacteria is present or not. New methylene blue stain. Okay. Next is flagella stain. Flagella stain, there are two types, wet mount flagella stain and leaf cell flagella stain. Stains for spirochetes, Fontana stain for Borrelia, Livadity stain for tissue selection, and silver impregnation for tissue selection. Other is capsular staining, that is Indian ink method. This is important. Okay. It is a negative staining. This is important. It is a negative staining or Negrosin stain. Everything gets stained instead of capsule. Only capsule is not stained, other all is stained. So that is a simple way to remember. The capsule is not stained. Everything else is stained in Indian ink method. That's why it is said negative staining. Okay. Or Negrosin stain. It is used for Cryptococcus neoformans. And this is the shapes or morphology of the bacteria. Staphylococcus aureus in clusters, Clostridium botulinum, and Klebsiella, Clostridium tetani, Streptococcus pneumonia. This is Diplococci. Diplococci means there is two cocci. Bordetella pertussis, Neisseria gonorrhea, that is also Diplococci. And Neisseria gonorrhea, another version. E. coli or Salmonella, Vibrio cholerae, Streptococcus pyogenes in chains, Bacillus cereus, rod-shaped, Helicobacter pylori, and Treponema pallidum. So, Treponema pallidum is corkscrew motility. So, let's go through culture medias. You just have to read this, and this is not important. There is solid media, liquid media, and semi-solid media. Based on growth requirement, there is simple media, which is minimum requirement for bacterial growth, like peptone, water, and nutrient agar. Enriched media, blood, serum, or egg. Sorry for these kinds of mistakes. I don't know how it happened. Um, if it's bothering you, please let me know in comment section. So I'll try to solve this in my next classes. Sorry for that. So next is enrichment media. So enrichment media contains blood, serum, or egg. And the example is blood agar, chocolate agar, Robertson's cooked meat medium, Loeffler's serum slope, and telluride blood agar. The other one is enrichment media. Selective media, it means CANPAAEMB, XLD, hectanoenteric agar, McCongie, TM agars. All these are selective medias. You just have to go through this. Differential media, transport media, indicator media, and anaerobic media. The next is synthetic media, which is chocolate agar. It is usually for the isolation of fastidious pathogens, Haemophilus influenza and Neisseria species. This is the very important one. This is chocolate agar is mostly used for these two species, that is Haemophilus influenza and Neisseria. It is prepared by heating the blood agar. First, the blood agar is heated, which in turn ruptures the RBC, and the rupturing of the RBC gives it a chocolate color and releases nutrients that aid in the growth of fastidious bacteria. The next is blood agar. Blood agar, it is prepared by autoclaving nutrient agar and pouring blood. It is prepared by autoclaving nutrient agar first and then pouring blood into it. Okay. So this one is important. Okay. So this blood agar shows three types of hemolysis, that is alpha hemolysis, beta hemolysis, and gamma hemolysis. So beta hemolysis is the complete hemolysis because it has a clear zone of lysis around the colonies. Okay, shows the clear zone of lysis around the colonies. The example is Streptococcus pyogenes and Streptococcus agalactiae. And the other one is alpha hemolysis. Alpha hemolysis is the partial hemolysis, and it shows a greenish gray or brownish zone of lysis around the colonies. And the example is Streptococcus pneumoniae. And the other one is gamma hemolysis. Gamma hemolysis means no hemolysis. And the example is Enterococcus fecalis. So the complete hemolysis is beta hemolysis. The partial hemolysis is alpha hemolysis, And the no hemolysis one is gamma hemolysis. So this is very important. Complete hemolysis means beta, partial hemolysis means alpha, no hemolysis means gamma. Okay. Next is enrichment growths, alkaline peptone water for Vibrio cholerae, selenite F growth, tetrathionate growth for Salmonella, gram-negative growth for the recovery of Salmonella and Shigella from clinical specimens, differential media. Okay. Differential media clad is used for E. coli and the color of E. coli in clad is yellow. Okay. And Mekangi agar. TCBS is for Vibrio, that is also important. Okay. So the clad is for E. coli and TCBS is for Vibrio. And in clad, the color of E. coli will be yellow. That is also important. So TCBS is for Vibrio. And in Mekangi agar is usually used for gram-negative bacteria. Okay, so Mekangi agar is usually used for gram-negative bacteria to differentiate lactose fermenters and non-lactose fermenters. If it's lactose fermenters, then it gives a pinkish color. If it's non-lactose fermenters, it gives a transparent color. Okay, so it is usually used for gram-negative bacteria, but there is an important point. It diminished gram-positive bacteria except Staphylococcus. So Staphylococcus aureus usually grows in Mekangi agar, but all other gram-positive bacteria is diminished by Mekangi agar. Okay, but the only one which is not diminished is Staphylococcus, that is Staphylococcus aureus. So this is important one. The only gram-positive bacteria that grows in Mekangi agar is Staphylococcus. The next is XLD. XLD is used for Salmonella and Shigella. If it's black, then it means Salmonella. And if it's colorless, then it is Shigella. Okay. So this is also important one. Okay. XLD, black means it's Salmonella and colorless means Shigella. This is an important question. What can be added in media to make it selective for gram positive? That is colistin, nalidixic acid, phenyl ethyl alcohol. What can be added in media to make it selective for gram negative? It is crystal violet, bile salt, eosin, and methylene blue. Okay, so this two is important. Next is transport media. There is towards medium, Pike's medium, sputum, Amis media. It's for use for Neisseria. Carrie Blair medium is used for stool, and buffer glycerol medium. They will not promote the growth. They will just support them to survive. So the important one is XLD. I have said that XLD is to differentiate Salmonella and Shigella. If it's black means it's Salmonella and it's colorless means it's Shigella. Then Mekangi differentiates lactose fermenters and non-lactose fermenters. The only gram-positive bacteria that grows in Mekangi is Staphylococcus aureus and TCBS is used for Vibrio. So these are the important ones. So I hope this class was good, and it made you guys understand about the topics. And I will catch you in the next class. If you have any suggestions, you can just comment below. And please subscribe my channel and support me for more classes. Thank you.